Great. So good afternoon and welcome everybody to our 2021 PhD Showcase webinar. My name is Niamh McCabe and I'm Translational Research Coordinator here at the Irish Cancer Society. And today I have the privilege of chairing this exciting showcase where we'll be hearing from two of our most recent translational biomedical research PhD students. A PhD is never easy at the best of times, but given the upheavals they've had to face over the past 18 months, we're so proud of the work that they've accomplished so far. So for our format today, first up, we will have a presentation by Rebecca Sheridan. Rebecca is just coming to the end of her second year of her PhD project, so near the halfway point. Rebecca graduated with a BSc in genetics from UCD in 2018 and went on to carry out a master's in biotechnology, bioinformatics and biobusiness at the University of Aberdeen. Rebecca was awarded an Irish Cancer Society funded PhD scholarship in 2019, so is currently just finishing the second year of her PhD in the UCD Conway Institute under the supervision of Associate Professor Margaret McGee. Rebecca's project is focusing on improving multiple myeloma treatment, so I can't wait to hear what progress Rebecca has made so far. We'll then take a few questions from the audience. If you have any, please put them in the Q&A box below. And these can be about Rebecca's project, about Rebecca's experience of her PhD so far, or any other relevant questions. So yeah, we'll try to get to them all then. After this, I will then introduce Adele, who will present on her project. And again, we'll have a Q&A session after that presentation. So without further ado, here is Rebecca.
Perfect. I'll just share my screen. Okay, so firstly, I just want to say thank you for everyone to come and listen to what my work has been so far. And also I want to give a huge thank you to the Irish Cancer Society for inviting me to come speak and also for funding the research that I'm presenting today. So Niamh has given an introduction, so I'm Rebecca. I'm carrying out my PhD in UCD's Conway Institute under the supervision of Associate Professor Margaret McGee with a co-supervisor of Dr. David Gomez-Matalons. So I'm nearing the end of the second year of my PhD. So I'm just going to go through some of the work that I have done up to now on my project that is titled The Characterization of Extracellular Vesicles Content and Function within the Tumour Microenvironment. And this is with a special interest in multiple myeloma as a disorder. So basically, I'm just going to go through what is multiple myeloma. So multiple myeloma is a hematological malignancy. So that means that it's a blood disorder, specifically a blood cancer. And it occurs in the immune cells known as plasma cells, which live in the red bone marrow of all of your bones. So on the top here, you can see what the function of a normal plasma cell is. So generally, plasma cells are quite beneficial for your body to have. They're in small numbers within your bone marrow, but they have an important function. So when your body comes under any type of infectious attack or notices a foreign substance presence, these plasma cells become alerted and they produce antibodies that recognize an attack and the infection, which in turn gives you an immune response and helps your body fight the infection. But unfortunately, when multiple myeloma occurs, defaults happen within these cells. Normally, genetic manipulations occur, which results in the cells being able to multiply out of control, leading to a much larger number of these cells being present within your bone marrow. That means that they're outnumbering the healthy cells within your bone marrow and dwindling down the positive immune reactions that they can cause. Additionally, they release a lot of antibodies, but these antibodies, unlike the healthy plasma cells antibodies, have no function. They do not respond to any infectious attack or foreign substance, and all they do is dilute down the positive antibodies in which the normal cells have released. This leads to a reduced immune response, which is a major problem with multiple myeloma. So my project is mainly focusing on the extracellular vesicles associated with multiple myeloma. So what are extracellular vesicles? These are small particles that are released from every cell within your body, but we do know that they are released at a much greater rate from abnormal cells, including those of multiple myeloma cells. So these small particles, as you can see in the diagram here, are just small bubbles which are released from the cells. Inside of these, there's cell components and genetic material, which are like a snapshot of the cell of origin. So they give a bit of insight into what is currently happening in that cell. So if it's a multiple myeloma cell, an abnormal cell, it will have different components packaged into it than a normal plasma cell. And on the outside of these, it contains various cell recognizable components. And these components are important because they allow the EVs to decipher which cells, healthy cells in the bone marrow, they should enter into and release their contents into. And in this way, it functions as a form of communication. So if we look at this diagram down here, a multiple myeloma cell is present in the bone marrow and it releases these EVs. So say this is the bone marrow within your hip. When these EVs are released, they have the option of being taken up, if it has the correct external components, into a cell within the bone marrow that the multiple myeloma cell is presently in. Then it can release its contents and inform the cell of how to behave best to benefit the myeloma cell. But these EVs can also move into the bloodstream and migrate to a different bone marrow. So say one in your shoulder bone marrow. And then if the lock and key mechanism works and it's recognized, it can enter a cell there. And there it informs these cells of how to behave in a way that benefits a multiple myeloma cell if it was to migrate to this location. So in that way, we think it aids the development of the disease. So what I'm doing is I'm working with human blood and bone marrow. So I'm going to be looking at the plasma cells and isolating EVs from these. So I'm lucky enough to be able to acquire samples from both normal and multiple myeloma patients through a collaboration with the Mater University Hospital with a consultant hematologist there, Professor Peter O'Gorman. When we get these samples, we process them to isolate the white blood cells, which is where the plasma cells are, and the liquid component of the samples. So essentially what we do is we get blood and we put it on top of a very, very dense liquid, which means the blood is much lighter and will float on top. Then we spin this tube at a very high speed and it causes separation of the blood components. We have no interest in the red blood cells and they're down the bottom, but it's the PBMC layer, which is the white blood cells, and the plasma layer, which is the liquid component and contains all the EVs, which we are interested in. So what am I doing to solve the problems that I've outlined? Well, I'm currently working with two different types of cell lines. A primary cell line, which are from patient samples, so the blood and bone marrow I've just described, and immortalized cell lines. Immortalized cell lines were once a primary cell, but it's been manipulated and high levels of mutations have been introduced to allow them to grow in a lab setting immortally. So these cells are very beneficial for standard protocols within the lab, but of course they're not an exact example of what a primary cell is, so it's better to be able to use both. So I'm isolating EVs from the multiple myeloma cell lines to gain a better understanding of their components and function, and also to develop some techniques. Then what I'm currently doing now is isolating multiple myeloma derived EVs from patients' blood and bone marrow samples. And from this, I'll be able to compare the two groups, the normals and the multiple myeloma patients' EVs, to decipher if there's any cancer-causing components. So components that might be in a much higher quantity in, say, the multiple myeloma group than the normal group. So this is just a bit of my research. I'm not going to go too in depth, but we looked at if multiple myeloma EVs had the ability to alter immune cell function, so other immune cells in the body. So we isolated EVs from multiple myeloma cell lines and made them a green, fluorescent green color. So as you can see here, the internal of the EVs are fluorescently green. Then we incubate them together. So we just add them to a plate that contains human immune cells, and we just leave them there for a day and check if the immune cells readily or happily take up these EVs. If these EVs are happily taken into the cell, they release their internal components, including that of the green fluorescence, resulting in the immune cell turning green or green fluorescent under a laser light. Additionally, what we're saying is that these EVs have an effect on the cells that they're taken up by. So we wanted to check if that was the case. So we decided to examine if the cells which had taken up the EVs behaved any differently. So the results we got was that these are increasing quantities of EVs added. So as you can see, that the more EVs that are added to the cells, the more the cells take them up, which suggests that the cells are happy to take up the EVs produced by multiple myeloma cells. When we checked to see if there was a difference in the behavior or the release of these components, we looked at two, MMP9 and IL-6. Both of these are generally released in an immune response, but they're bad components to be released in a multiple myeloma prognosis. This is due to the fact that they help the progression of the disease when they're present in high numbers. So as you can see here, more EVs added resulted in a greater release of these components, suggesting that these EVs alter the immune function to make it a nicer environment for the multiple myeloma cells. Additionally, as I mentioned, I'm working with plasma cells and EVs from human blood and bone marrow. So with the cells, we decided we want to characterize the components that are decorated on the outside. Just as I explained, there's components, the keys on the outside of the EVs. So what we found is certain markers are present in a much higher rate in all multiple myeloma patients, or the multiple myeloma patients that we've received, compared to that of normal groups. So this is good to be able to separate out the two groups into multiple myeloma and normal control. Additionally, we took the plasma, or the liquid component, that contained all the EVs in the blood. So it's important to note that all these EVs, they're not just from the multiple myeloma cells, they're from all the cells within the body. But as mentioned, there's much higher numbers of multiple myeloma cells when the disease is active, and also they release a much higher number of EVs. So the likelihood is that a large quantity of these are going to be multiple myeloma associated. What we're going to do is characterize the components on the outside, similar to what we have done here with the cells. But additionally, we're going to use specialized technologies to check the protein or the cell components and the genetic components that are packaged inside these, so that we compare the two groups of normal controls and multiple myeloma patients, and therefore identify differences between them. So what I'm planning on doing next, I still have 2 years left. So I'm going to continue the sample collections of both the multiple myeloma and normal controls. I'm going to analyse physical characteristics similar to what I've just explained there of all the samples. So I should have 20 patients and 20 normals at least. And then through this, I'm hoping to be able to identify factors associated with multiple myeloma progression. And this may lead to better detection methods, earlier treatment, and overall improvement of the patients. But it's important to note that my study is very early on in this process. So no direct detection method will be developed for mine, but I'm hoping to be able to provide a large amount of data that other research can build off of. So finally, I just wanted to cover one thing that is very important to the Irish Cancer Society, and that's the public patient involvement. So unfortunately, I am only coming to the second year of my PhD, and for a large majority of that, we have been in the COVID-19 pandemic. So I've been limited in as a lot of the PPI that I could be involved in, but I have still had the opportunity, and in the future, I'm going to continue this. So the biggest part of PPI for me would be that I have a patient researcher mentorship program. So that is every 3 to 6 months, I meet up with a group of 3 patients and a member of the Multiple Myeloma Ireland Association. And we sit down and we discuss my research plan, the data that I've got, how I should share this, and how best to reach the public with my information, with my results and data. And we are hoping to have lab visits as well, but unfortunately, due to COVID, that has been limited. Additionally, we do public talks like this. So like the showcase that we're in now, and UCD additionally have an Engage seminar, which is a series of PhD talks that is solely focused on a public audience, and I will be participating in that. And then also information sharing and getting involved. So the Irish Association of Cancer Research has an award that's primarily based on your ability to inform the public about your research, which I plan on partaking in this year. I've attended the Multiple Myeloma Ireland Patient and Care Information Day, which has given me the opportunity to talk to both patients and healthcare workers heavily involved in multiple myeloma treatment, and has given me a great insight into the other side of the work being done. And then also I plan to be involved in World Cancer Day in UCD and Patients' Voice in Cancer Research once things get up and running again. So thank you all for listening, and if you have any questions, I'd be more than happy to answer.
That's great, Rebecca. Thanks so much for your presentation there. So like that, I have a few questions there from the attendees so far. But I might start with one of my own if that's okay. So Rebecca, you gave a great explanation of the value of patient blood samples in your work, that were provided by Peter O'Gorman through that collaboration, and you explained how you process them. Just from a logistical point of view, I'd love if you could take us through like how a patient attending a clinic in the matter provides their blood sample and it reaches you in UCD. This must be like time sensitive.
Yeah, so every Tuesday, we work with the registrar over in the matter for in the hematology department. So every Tuesday, she takes bone marrow samples from patients not only with multiple myeloma but just hematological malignancies, so blood disorders. But a lot of them do have either suspected early stage myeloma, which would be MGUS, so it's the non-malignant stage, but it's leading into the kind of cancer stage of myeloma and myeloma patients themselves. So she addresses them before they get their marrow taken and provides them with a patient information sheet that explains what we're doing and what's happening. And then they're given time to think about it. They get the option if they want to sign the consent form or not. If they sign the consent form, whilst their bone marrow is being aspirated for their medical procedures, they take a little bit extra from myself, and then they go to a phlebotomist who just takes the blood, which they also consent to. And then at about 12 o'clock, I get a text message saying I need to be in the matter at 2 o'clock to come pick up the blood. And then I bring it back to UCD, and then I process it that day. So it's a long enough procedure, long enough day to get it done, but there's a lot of steps to it. So
Yeah,
yeah.
absolutely. No, it shows the benefit, of course, of the patient involvement and how important that is to research.
Mm.
Um, so that's brilliant. So I like that. So more of a science question we have in here. So Rebecca, so you expose immune cells to EVs. The more EVs they're exposed to, the more EVs taken up by the immune cells, resulting in the more dysfunction of immune cells shown by increased MMP9 and IL-6 levels, and that can be associated with poor prognosis and progression of disease. So does that mean that levels of EV can be used as a measure for staging disease? That advanced disease has higher EV levels than early stage, or if early stage disease have high levels of EV, is that an indication of an aggressive disease?
So I think it'd be very difficult to say specifically if it would be increased EVs, because we don't know exactly how many EVs an early stage myeloma cell releases compared to a later stage. We just know that the myeloma cells release a lot more EVs.
Hmm.
But I think the main thing is later stage myeloma cells, it's what's packaged into those EVs that drives that release and the increase of the components that are going to lead to a more beneficial environment and a worse prognosis for the patient. So it's more so the components. So that's why we're interested in identifying what is actually inside these EVs.
Yeah,
And
yeah,
luckily, we do get the opportunity to receive some of the patient's medical data. Nothing outside of what would be important for our research, but we'll be told whether they have developed any type of drug resistance, or if they have been treated before, if they're late stage, have they had a stem cell transplant. And all of this, then once we've collected all our data, it means that we can cross compare kind of different subgroups as well. And hopefully
absolutely.
we'll be able to identify different components, not just between the normals and the myelomas, but within the myelomas themselves. But obviously that all just depends on how many of each, like kind of smaller early newly diagnosed and like drug resistant patients that we get, because obviously we're very lucky for any patient to donate to us, so we
Yeah,
just whatever we're given we're going to work with.
yeah.
So we're hoping that we get enough of a couple kind of subgroups that we can kind of do a bit of a comparison there as well.
Absolutely. So like that you can retrospectively then look at the differences that you'd already identified and then see how that resulted in clinical presentation later on.
Yeah.
Brilliant. We have another sciencey one here. So Rebecca, you're looking at EVs to identify cancer-causing components. So that would be the parts in the EVs, I suppose, that you were saying. So then, are these specific to EVs, or are they representative of the whole tumor? So you wouldn't be the goal wouldn't be necessarily to target the EVs themselves. It might be to so the EVs would be a representative measure or biomarker to show what you could target in the tumor. Would that be correct?
It would be correct, but then there's also the concept that if there is something that's heavily upregulated in the EVs, well, if you can block that reaction while they're migrating in your blood, you can stop them from progressing the disease, and the fact that they're known to cause to set up like new environments for the movement of the disease and stuff like that. So if you can block the EVs
Yeah.
while they're migrating, so say like well, there's a marker that there's specifically a drug targeted towards, but we know that this marker is actually highly expressed on EVs as well. So there's the question of, is it possible that the EVs are actually sequestering the drug, and that's why resistance is occurring and things like that.
Okay, so it's EV specific.
So it's a bit of both. Like it could work in either direction. We just want to find markers, and then we can decipher what's best in the future.
Absolutely.
But yeah.
And we have another question in. Hi, Rebecca. Have you done any work on myelofibrosis?
No, none.
No, that hasn't fit in.
At all.
Yeah, you've enough to be doing at the moment anyway with your plans. So it'd be interesting to see further down the line what it could lead to.
Yeah.
And one final question then. So Rebecca, what do you enjoy most about doing a PhD? I mean, I'm sure we can guess what was most challenging over the past 2 years for yourself with COVID and everything else. But so it's nice to maybe look at one of the more positive aspects of your PhD.
I'd say, well, obviously getting to do the research, and I like the aspect that I get to collaborate with the matter, and it kind of makes a different environment to it. But I also think a big part is like there is a social side to being in a PhD as well, and it's great the format of in Conway that if you have a research question, you don't just have to go to your group. There's a lot of people around that you can kind of bounce ideas off, and I think that's quite nice as well.
Brilliant. So it's a really collaborative atmosphere, and you feel supported then.
Yeah.
Brilliant. That's great to hear. And Rebecca, thanks so much for taking the time to present to us today, and we'll let you go now.
Perfect. Thank you.
Thanks, Rebecca. That was brilliant. I think gave us all such an insight into your experience with PhD so far. And I think I speak for everyone when I say I've learned so much about multiple myeloma from your presentation today. But next we have another PhD student, Adele Connor. So Adele completed a BSc in Biology and Chemistry at Maynooth University in 2014, followed by a Master's in Biomedical Science at University of Ulster in 2015. Following this, Adele then worked as a research assistant in Professor Antoinette Perry's lab in UCD. Adele then decided to pursue a PhD examining the cell biology of ovarian cancer. Adele was awarded the Irish Cancer Society PhD Scholarship in 2020 and commenced her studies this January in 2021. So take it away, Adele.
Yeah, no problem. I'll just share my screen here now. Yep. So yeah, hi everybody. As Niamh said, my name is Adele Connor and I'm a first year PhD student funded by the Irish Cancer Society. And I'd just like to start off by saying a massive thank you for giving me the opportunity to speak to you today about my PhD project, which will focus on zooming in on ovarian cancer. So there's a bit of a background about ovarian cancer to begin with. So ovarian cancer is the 4th most common cause of cancer death in women in Ireland. And sadly, it has a 5-year survival rate of only 37%. Now, one of the major reasons for such a low survival rate is the fact that 3/4 of women present with advanced stage disease that is already spread around the body. Now, although 85% of patients will initially respond to treatment, this can be pretty short-lived and resistance will quickly ensue, which is why we desperately need new treatment options for these patients. However, what we currently don't understand is how these different tumours, so the primary and the metastatic, respond to therapies within the one patient, which is what I'm hoping to address with my project. So what am I doing and why? So the first question I have is, what are the differences between the primary and the metastatic tumours within the one patient? And secondly, then, how do these differences impact treatment response for that patient? So in order to address this, I'm planning to look at 3 things. So firstly, I want to define the observable differences between the primary and the metastatic tumours. So what that means is looking at the structure of the tumour and also looking at the number of cells that comprise that tumour. Secondly, then, I want to profile the tumour environment. So a tumour is not just an isolated group of cancer cells. In fact, it interacts with the different cells inside our body, including our immune cells. And tumours can then be classified based on the number of immune cells that surround it. So what you can see in the little image here, so your pink cells are your cancer cells, and that forms your tumour. And then your blue cells here are your immune cells. So this would be known as a hot tumour because it has a lot of immune cells surrounding it. We can also have cold tumours like the one we see here. So as you can see, there are a lot less immune cells surrounding this tumour, so it's known as a cold tumour. Lastly, then we can have immune excluded. So these are tumors that have no immune cells surrounding them at all. And the reason why this is important is because hot tumors generally respond better to immunotherapies compared to cold or immune excluded tumors. So lastly, then, I want to assess how each tumor responds to combination therapies. So I plan to treat these tumors with immunotherapies and to then repeat steps 1 and 2 again. So I want to now look at the structure of the tumor and the number of cells and try to understand which tumors have responded and which tumors didn't, and to understand if these therapies has had an effect on the structure. I then again want to look at the tumor environment and to see which tumors responded, which tumors didn't, and then to see if it has had an effect on the number of immune cells surrounding the tumors as well. Now, the reason why I want to do all of this is that I'm hoping it can lead to personalized, tailored treatment for patients. So how do I plan to do this? So patients who present with advanced stage ovarian cancer will undergo surgery to have their tumors removed. These patients can then provide informed consent to then donate those tumors to my research. And then I will work closely with Professor Donald Brennan, who is a gynecological oncologist at the Mater Hospital, who will perform these surgeries and then give those tumor samples to me. I can then bring these tumors back to the lab and continue to grow them. Once I have them growing, I can then add on fluorescent dyes, which will bind to protein markers that are inside these tumors. The next step then is to look at these under the microscope. So I want to take each of the individual tumors, so the primary tumor and each of the individual metastatic tumors from the one patient, and to look at these closely under a microscope. This microscope then allows me to take pictures of what these tumors look like. And what allows me to visualize these tumors are these fluorescent dyes. So I have numerous different fluorescent dyes that all bind to different markers. And if I add on a marker to a tumor, I add on a fluorescent dye to a tumor that does not possess the marker of interest, then this dye cannot bind and will not fluoresce. And therefore I'll be left with a dark image. However, if I add this dye onto a tumor that does contain the marker of interest, the dye can bind and it will fluoresce, and I'll be left with a bright image. So this shows me part of the structure of this tumor. And this is how I plan to compare and contrast the primary and the metastatic tumors from the one patient. These dyes will help me paint a picture of the structure and composition of each individual tumor. So what has my progress been so far? So the PhD is a 4-year journey, and I am currently coming to the end of my first year. So it's still very early days, and I do have a long way to go. But what I've been focused on so far is looking at these different fluorescent dyes and making sure that they work. So to do this, I've grown ovarian cancer cells, which can be bought in. They're less precious and they're easier to work with than patient tumor samples. So I've grown these ovarian cancer cells, I've added on the dyes, and I've then taken pictures. So what you can see here in this picture is an example of 2 different dyes. So the blue dye attaches to the DNA inside this cell, which lives inside the brain of the cell. The green dye then attaches to the skeleton of this cell. And so together, these dyes can show me the structure and the size of this cell. Another thing then I've been working on is the development of my patient advisory committee. So to do this, I've been working with the women of Ovacare, which is an ovarian cancer community. And the idea of this committee is that they can provide guidance and feedback on my project from a patient's perspective. So I've worked with this group since the application stage of my project, and they were able to provide great insight into the development of my patient involvement plan. Currently, we're working on developing our terms of reference for the committee. So this will just outline how the committee will run and what the patients can expect, but also what I can expect from the group. Lastly, then they also provided some feedback on an infographic that myself and Romina Silva, which is another Irish Cancer Society PhD student, created around raising awareness for ovarian cancer for World Ovarian Cancer Day. So I've absolutely loved working with this group, and I'm really excited to see where the committee develops to over the next 3 years. So what are my next steps then? Although talking through my project here today was pretty quick and easy, and talking about taking the tumor samples and growing them in the lab, I was able to go through that pretty quick, but I actually currently don't have the skills necessary to do this, which is why I plan to travel to the University of Leicester to Professor Catherine Pritchard's lab, whose team are experts in this field. So I will spend 1 month here learning how to take tumor samples from a patient and to grow them in a lab environment. So I'm really looking forward to going there and then to be able to bring that skill back to UCD. And then for the last 3 years of my project then, I plan to recruit 5 patients who will be able to donate tumor samples. And then I plan to look at these tumors, so look at the structure, look at the tumor environment, and to treat them with combination therapies. So with the idea that this could result in rapid analysis of therapeutic responses that could then be returned to the clinic as actionable information. So if we can understand the differences between the primary and the metastatic tumors, this could then provide targeted personalized therapy for patients, which would ensure that the patients who stand to benefit will receive the right treatment, but also that those patients that won't benefit will be spared the pains of ineffective treatment. So that is all from me today. I'd just like to say a massive thank you to my 3 supervisors, so Associate Professor Antoinette Perry, Professor Jeremy Simpson, and Associate Professor Joanne Lysett. I want to say thank you to Professor Donald Brennan, who is the gynecological oncologist at the Mater Hospital, who will perform the surgeries and provide me with the tumour samples. And then a special thank you to Dr. Sharon O'Toole and the Women of Ovacare. They have been absolutely incredible in helping me develop the Patient Advisory Committee, and public and patient involvement in research is a bit of a new step for me. So they've been really supportive. So I'm very excited to see where that group goes to. And then thank you to all the lab members in the Cancer Biology and Therapeutics Lab, the whole cell screening team, and all of the group of the Perry Lab as well. So thank you very much for listening.
That was great, Adele. Thanks so much. So I'll start with a question that came in there. So they are asking for you to explain what the difference between a primary and a metastatic tumor is, if that would be okay.
Yeah, so the primary tumor is where the cancer will originate first. So in ovarian cancer, that's in the ovary, the fallopian tubes. That's where you'll see your first tumor. And what a metastatic tumor is then is if that cancer then manages to spread around the body, it will spread to a different site inside the body. So that's what your metastatic tumors are. So yeah.
Brilliant, thanks. And following on from that, I have another question here. So it was saying that, as far as from my understanding anyway, you're looking at the differences between, say, a primary tumor and tumors at sites of metastasis within the same patient. Were you saying that even within the same patient, between the primary and site of metastasis, one tumor could be hot and another cold?
Yeah, so you could see very different tumors inside the one patient. So yeah, like so potentially one tumor might respond to the treatment, but another tumor could have a very different environment, very different structure, and it might not respond to treatment. So that could be potentially why patients might not respond to treatment overall, because one tumor responds, but the other doesn't. So I'm trying to do a very highly personalized approach by looking at all the tumors within the one patient, trying to assess what those tumors look like, which ones respond, which ones don't, to be able to try and understand why ones responded and why ones didn't. And then ideally, down the line, if we could then use that as a way to then advise on treatment for a patient. So if all the tumors respond, that patient might benefit from a specific treatment, but if they don't, well then that treatment might not be useful for that patient. But yeah, different tumors could be very different across the one patient.
Absolutely. So that's showing, I suppose, how complex cancer treatments can have to be.
Yeah, exactly. Yeah.
So like that, it shows that, not only will it differ greatly between 2 ovarian cancer patients, but even within the same patient.
Yeah, which makes it very difficult to treat then as well, and makes every person individual. Yeah.
Absolutely. So we have a question there around, so I'll go on to your ovarian cancer patient section. So somebody's asking, how will you recruit the 5 ovarian cancer patients?
So Professor Donald Brennan at the Mater Hospital, so he's a gynecological oncologist there. So he already had ethics in place in order to be able to talk to patients there who present with, specifically I'm looking at patients who have advanced stage high-grade serous ovarian cancer. So as they present to him, he will be able to provide them with information about the research I'm doing, and give them the option that if they want to donate their tumor samples to the research, they can do that. So there'll be full information about what the project, what will happen, the research that's involved, and then they can provide consent for that. So that's how that will work.
Brilliant. I know you gave a really specific overview, or really simplified overview of the explant treatment and everything, but obviously it's much more complicated than how you described today. So would you expect to require more patients than that, or is it more that you'd use other samples to kind of hone your craft when it comes to the explant?
Yeah, so first? I don't know a huge amount about the explants. Yeah, as you said, I need to go over to Leicester to learn exactly how to do that, and I was meant to go in May, and then I was meant to go in June, and COVID has pushed it, so it's booked for September, which is very exciting. But yeah, so I'm hoping the 5 patients is what I plan to do, just in the time frame that I have for my PhD, and to be able to look at that in depth, I can only do the 5 patients. But I think maybe when I return from Leicester, there might be maybe towards the end of the year a patient that, just to try and figure out like the dyes, to work on them, the doses that I need and stuff like that. But
Yeah.
I'm maybe a couple of pages at the end of the year that I might need to take the tumors and exactly hone the craft to be able to then have a cohort of 5 individuals that I can have results from.
Absolutely, but you're going to experts in the field by going to Leicester anyway, so
Exactly,
yes,
exactly. I'm hoping they'll be able to show me the way on that.
Brilliant. And then we have a few questions about your experience of a PhD as well.
Yeah.
So we have a question from an attendee saying that I'm about to begin my undergrad studies and I'm hoping to do a PhD in cancer research. Do you have any advice for doing or applying for a PhD project?
Okay, so I think one of the biggest things I think to understand is that there is no one way to go about it. Like everybody that I work with here has had very different paths to get here. I suppose it's making contact with maybe the lecturers that you work with, if there's a lecturer in your university that's doing something that you're interested in, make sure you make contact with them. If you can do your 4th year project potentially with them, that's a great way to kind of understand, like meet the lab group, understand, and then use the lab groups that you can get into. They can provide really good advice. Another way is, because obviously a PhD is a big undertaking, what you could do first is, if you didn't want to go straight into that, you could do a research assistant position. That's what I did. So I did my undergrad, did a master's. I actually went traveling for a bit after that and then came back and did research assistant for 2 years. And that's what really kind of, I was able to talk to the girls here, understand the pathways that they did. There are different funding bodies that you can do, and there's different lecturers you can do it with as well. So I think just make contacts with as many people as you can. I'm also happy if they wanted to contact me afterwards, if they wanted to chat, I'm happy to do that. I can pass my email address on if they wanted to, but just there's no one way to do it.
Yeah, no, absolutely. I'd agree with that. And I think you hit the nail on the head there as well, that, it is a big undertaking doing a PhD. So it is worth doing a bit of research and, knowing that it will be the right fit for both of you and not just jumping into a project. Because if you don't have the same motivation to pursue it, I think it makes it so much harder if it's not something that you're really interested in as well.
Exactly. And the lab group, I think, is such an important part of this. I'm lucky to be part of 2 incredible lab groups here, and I get on with all of them, and that makes such a big difference. That like if you're having a bad day, there's someone there to help. There's always someone around to help. So I think that's a big part of it as well. If you could get into the group and kind of see if it's a good fit, then too.
Absolutely. And of course, if there's any other questions like that, you can email them to research@irishcancer.ie, and we'd be happy to help give any advice we can as well. And we have another question there as well about PPI in your project. PPI is a really important part of research, but it can be hard for people to know where to start. What advice would you give to other researchers on how to kickstart PPI and integrate it into their research?
Yeah, so as I said, it is very new for me as well. When I was doing the application for the Irish Cancer Society, it was a bit of like, I'm not exactly sure what I need to do here. So there's lots of there's stuff online and there's papers that you can read that was very helpful. There's the Patient Voice in Cancer Research in UCD. They have stuff there as well to kind of give you tips on what you should do. But one of the things that I did was I looked up, because mine was in ovarian cancer. So I just found different ovarian cancer charities or groups that were in Ireland, and I just emailed them. I contacted people on Twitter and just kind of explained what I was doing, said that I was really interested in doing some patient involvement, that I wasn't really sure how to go about this, but if they had any help. And luckily, Sharon O'Toole got back to me, and she's been really, really incredible. So she knows her stuff in terms of like, there's the Overcare group there already. So I think if you can contact a charity that way, there's a really good way that it's already established. They know what they're doing and they can help you then too. I think that's the best way. That it worked out for me anyway.
Absolutely. I think that's great advice. We have another message in there to say that it's great to see this work for such a challenging diagnosis being done, and well done to you then. So I'll finish off with one final question.
Yeah.
Like that, you're only in the first year of your PhD. What has been the most surprising aspect so far for you? And what are you looking forward to going into your final 3 years now?
Very good question. I think the most surprising aspect, so obviously I did a research assistant for 2 years and absolutely loved that. And I can remember Antoinette keeps telling me, like when I was doing the PhD, she's like, look, it is very different. And I kind of knew in the back of my head, I was like, yeah, no, it'll be a bit different, but I think it'll be fine. But I think I was surprised at how different it was. So in the research assistant, there was a group of us and we had a project. And if I couldn't do something, there was somebody else there that could pick that up. With the PhD, although you have a lot of support and you have group of people around you, if you're not doing it, it's not getting done. So that's, I think, one of the most surprising things. Like, yeah, like I kind of knew it was going to be different, but until I got into it, I didn't realize how different. And then I think the moving forward, is that what you were saying over the next?
Yeah, Yeah.
I'm so excited to get to Leicester because obviously it's been over a year in the planning, I think now. So I'm really excited to learn that technique and then bring that back. But then on top of that as well, I'm really excited to see where the patient advisory committee goes. I think that was one of my favorite things when I was applying for the PhD. Like it's a tough application process and there's a lot of papers that you're reading. And I think one of my favorite parts of that was to meet the women of Overcare. They were just so welcoming, so incredible, really lovely and really supportive. So I'm really excited to develop that and kind of really learn how to do the patient and public involvement a bit better and to, go really far with that one. So yeah.
Brilliant. That's great to have heard such positive experiences for both you and Rebecca of your PhDs so
Yeah.
far. Because there's a while left to go. So I think if your first and second years are hard, 4 years can seem like a very long time.
Yes, exactly. Yeah.
Great. So I think so that was brilliant. Thanks so much for presenting to us today and to Rebecca as well. So I'm going to wrap up our webinar today by saying thank you so much to everyone for joining us. I hope it was informative and enjoyable for everyone attending. A special thanks to both Rebecca and for Adele for presenting their research so far and for giving us such insightful talks. I can't wait to see where both your projects go over the next few years as well. And we'll be sure to have you back to see how it progresses from here. Finally, I want to thank Shauna, who is working away in the background, bringing these webinars to the public and managing all the logistics behind the scenes. So thank you so much, Shauna. And I hope you'll all join us for our next webinars. There'll be a few taking place in October, so be sure and look out for them as well. So thank you very much and have a lovely afternoon, everyone.
Thanks, everyone. Bye.